Good evening, everyone, and welcome to the CUBE's virtual seminar series that we're hosting in 2021. I'm happy to have Dr. Burton Lim join us and speak to the challenges and opportunities for bat fieldwork and research during the COVID-19 pandemic. I just want to let you know that we are recording, as noted at the start, this seminar, and it will be available on our CUBE's YouTube channel. There is an opportunity at the end to ask questions to Dr. Lim. You can do so via the chat, or you can unmute yourself and ask at the end of the presentation. Before we start off, I'd like to give an acknowledgement of the territory that we're on. I know people might be joining from different regions, but we're in the Queen's University Kingston area of Ontario, and it's situated on traditional Anishinaabe and Haudenosaunee territory, specifically the Queen's University Biological Station, which is about an hour north of Kingston, is situated on unceded Algonquin and Anishinaabe territory and is part of the Algonquin land claim by the Algonquins of Ontario, currently under negotiation with the federal government of Canada. Acknowledgement of these facts requires recognition of the pre-colonial history of this land and the peoples who lived here and continue to live here. The cultures and spiritualities of Indigenous peoples are connected to the land, and the land is an integral part of their ways of knowing and living. These knowledge systems are continually evolving in relation to the land and its other inhabitants, both human and other than human. The Kingston Indigenous community continues to reflect the area's Anishinaabe and Haudenosaunee roots. There is also a significant Métis community, and there are First Peoples from other nations across Turtle Island present here today. So thank you again for joining us. Before we go ahead, CUBE's director, Dr. Stephen Lougheed, will be introducing Burton to those here and those watching on YouTube later. Thank you.
Thank you, Sonia. It's my pleasure to welcome Dr. Burton Lim to our field station, at least virtually. I just extended an invitation for him to come up and explore a few bat caves, perhaps a little bit later on post-pandemic. Dr. Lim is currently Assistant Curator of Mammalogy in the Department of Natural History at the Royal Ontario Museum, where he's actually worked since completing his undergraduate degree. Burton did his BSc in zoology at the University of Toronto, an MSc in biology at York University. His PhD was done at the University of Toronto on the evolutionary history of the New World sheath-tailed bats of Central and South America. Burton also has a certificate in global journalism from the Munk School of Global Affairs. He's traveled to 28 countries, or perhaps more since the document that I saw, conducting fieldwork on mammals, focusing particularly on the evolution, biogeography, and biodiversity of tropical bats. Since 2011, Burton has been working with Operation Wallacea, monitoring biodiversity in Iwokrama Forest and Sorama Village in central Guyana, including bats in this work, tracking changes in the ecosystems there. He's also participating in the International Barcode of Life project to create a reference system for species identification and discovery of mammals. He had the exquisite opportunity to skin a blue whale for the Royal Ontario Museum. He may speak to that in the question period, I don't know. Burton has been chair of the board of directors of the North American Society for Bat Research. And in addition, he has been associate editor of Acta Chiropterologica. That one, I stumbled on that, forgive me, Burton. And this is an international journal on bat research, and this is since 2015. So with that, I'll turn it over to Burton. Thank you so much for agreeing to give us a talk, Burton.
Okay, thank you, Stephen. And let me just bear with me as I fiddle around getting my screen set up for sharing and presenting. Okay, now, are you seeing, I'm assuming, hoping that you see my presentation full screen?
Yes, we do.
Okay.
Thank you.
Excellent. And then I will continue on with my presentation. Yes, so the COVID pandemic has affected everyone throughout the world, and most tragically, the more than 4 million people that have died from the illness. So my presentation will talk about some of the challenges and opportunities for bat field work and research during these unusual times. So I'm assuming you can also see my background Zoom photograph. So this is of the Batcave exhibit at the Royal Ontario Museum where I work. So with the current provincial restriction that has closed the public side of the museum, this is one obvious impact on the ROM, is that we haven't had visitors in at the museum. But this will be lifted soon, in fact, this weekend, and we'll be opening up later this week. As Steve had mentioned, we also have a new whale exhibition opening up. So just a quick plug for people that are coming to Toronto. And if you can, drop by the museum to see not only the Batcave, but also the new whale exhibition that we'll be having on display. It will be going until March of next year. So you don't have to madly rush this weekend to come see it. Okay, so since the pandemic began 14 or 16 months ago, I've been essentially working from home like most of you, including spending a lot of time on the new whale exhibition I just mentioned. But tonight's presentation is on bats as related to the COVID pandemic. The picture on the top is a species, so I'm not sure if you see my cursor, but it is a species of horseshoe bat, which is the genus implicated as a natural reservoir of SARS-like coronaviruses. The image below is of the spherical SARS-CoV-2, one of the two severe acute respiratory syndrome viruses that has caused the highly contagious and deadly disease responsible for the current global pandemic. As you can see, the virus is surrounded by numerous spike proteins, which bind to the ACE2 receptor sites on the human cell. So this is how the virus enters the body and then uses the host to replicate more of itself to cause the disease. But a little bit of background first. Bats don't transmit SARS-like viruses directly to humans, but encroachment on wildlife and their natural habitats increases the probability of pathogens jumping to other species. There has been quite a bit of false information circulating around, especially on social media during this pandemic. One of the more infamous ones was the suggestion that the outbreak got started by people eating bat soup. But this was an old video on YouTube that predated the pandemic and was from another country, I believe Indonesia, not from China. But unfortunately, this led to the wrongful persecution of bats, including killing them in several other countries throughout the world where bat coronaviruses aren't actually found or known. Worldwide, there are about, actually more than 1,400 species of bats. So this is the second highest level of biodiversity in mammals, so after rodents. That makes up, so bats make up about 22% of the mammalian species diversity. And also bats are responsible for many essential ecosystem services, such as seed dispersal, flower pollination, insect control. They're also responsible for, in some cases, for fertilization, where people use guano. Bats are beneficial in many ways to the proper functioning of the environment. So bats in general have, sort of been vilified, again, sort of in the popular press, but in fact, biologically, they're very important cogs in the environment, in the ecosystem. Okay, I'll give you a bit of background information on bats and coronaviruses. So this is a study by Liu and colleagues from last year that was published in the scientific journal Lancet. And there's also been other studies as well that have sort of talked about bats and coronaviruses. But what they found was that the closest known coronavirus to SARS-CoV-2 occurs in the species of horseshoe bat from China. Again, hopefully you can follow my cursor there. However, there is a 4% difference in RNA between the bat virus and the human virus. To give you an idea what that means, Chimpanzees and humans are about 5% different in their genomes. So in this case, 4% is actually quite a bit between species, whether it's mammals or viruses. And a further study to estimate when the most recent common ancestor of the bat and human coronavirus existed was done by Bonnie and colleagues in this paper in Nature Microbiology last year. So this coalescent event dated back to about 1969, which means that the virus has been circulating unnoticed for the past 5 decades in some other unknown intermediate host before jumping to humans. Another difference is found in the coronavirus spike protein, which binds to the ACE2 receptor that I briefly discussed at the beginning in humans to gain entry into cells and affect the host. However, the bat virus, so the one marked in red with the arrow on the bottom there, has 5 amino acid residues that are different from the human virus that's at the top. So you can see that there's 5, I guess, blue boxes that hopefully you can see on the screen. That you can see where my cursor is. So those 5 amino acid residues are missing in the spike protein of the bat coronavirus. So this would make it difficult for the bat coronavirus to infect humans directly. So these 3 examples I just showed you are important pieces of information that tell us that the bat coronaviruses, or the coronaviruses in bats and humans are similar, but different enough that transmission was through a still unknown intermediate host. Okay, so that's just a very quick overview of the background information on the association between bats and coronaviruses. Now I'll get into the connection between ROM fieldwork and COVID-related research. So in 1989, we began preserving tissue samples at the ROM for genetic study, along with voucher specimens for research, primarily on the evolution and biodiversity of mammals. So over the last 3 decades, we've collected about 15,000 bat specimens from about 30 countries, highlighted throughout the world. But these are primarily from tropical places where most of the bat species diversity is found. And most of it, as you can see, is in South America and also Southeast Asia. And then the picture on the bottom is a picture from 2004 on a field trip to China. So this is a harp trap that is set across a creek in secondary forest. So harp trap, most people might be more familiar with mist nets, which are the same mist nets that are used, to survey birds. So we also use those for catching bats. But we've also started to use different, so I guess newer equipment or methodology. So this shows a harp trap that is used. So this usually is used better in sort of tight conditions where you sort of see like a flyaway, like a narrow flyaway such as this creek, or they're also very good at the entrances of caves where the cave sort of tunnels or narrows off. So this is a good way to collect a lot of bats that might be flying, coming in and out of a certain area, such as a cave. So this is a picture of a harp trap. So I mentioned China specifically because on this and other trips to China, we caught 4 species of bats that were new to science and which were reported in 2 of these papers. So that is another thing that my research is related to. It's looking at species discovery. So we have these repositories of specimens that we use for biodiversity and evolutionary studies. But now, as I'll talk a little bit more thoroughly, we can also use these tissue samples for other purposes that we had not thought about in the beginning, like even 10 years ago, about doing it for genomic studies, but also for looking at viruses also. Okay, so during these past 30 years of field work at the ROM that I've been a part of, The bat frozen tissue collection at the ROM has become an important resource and one of the best in the world. It has grown to represent 15 of the 21 families of bats, 120 of the 220 genera, and about 400 of the more than 1,400 species of bats. So the photos that you see here give you an idea of the range of morphological diversity in bats. So some bats have white stripes on their backs. And so this is the two-line sac-wing bat in the family Emballonuridae, which is what I did my PhD study on. This is a picture. Well, some bats also have just one line down the back, but also they have stripes on their faces. So this is a fruit-eating bat of the Trinidad big-eyed bat. And then other bats don't have any stripes, but some of them have these really huge ears and also these huge nose leaves on their face. So both of these are used for echolocation. So the ears receive back the echoes, but they also hear sounds of potential insect prey. And also the big elaborate nose leaves are used to direct the echolocation called signal as the bat flies around, or as the bat might be looking, finding or zeroing in on their prey that they're trying to catch. Okay, so in addition to having good geographic coverage and good taxonomic coverage, most of our tissue samples were originally quick frozen in the field in liquid nitrogen, which is really cold at minus 196 degrees Celsius. So basically, this is an instantaneous freeze. Once you drop, the tube of tissue in, it's quick frozen right away. So the importance of this is that the usual method is to preserve tissue in ethanol, but this makes it difficult to recover long fragments of DNA for newer technologies of whole genome sequencing, and also more difficult for the study of viruses, which have RNA, which is less stable than DNA. But of course, in addition, to trying to get liquid nitrogen where we're doing field work, so this can be a bit challenging at times, as is trying to lug around this tank around with you in the field, especially when you're in remote parts where you have to literally carry it in on your back to get into some of these places. But again, even though it's difficult, this is still the best way to preserve tissue for molecular study and long-term storage, especially for the newer genomic studies that are going on nowadays. Okay, so that was just sort of a quick introduction to bats and coronaviruses and some of the work, the field work, and how we preserve our tissue samples at the ROM. So for basically the second part of this presentation, I'll talk about specific examples, looking at two opportunities of collaborative research related to pandemic work that I've been doing, and then finish off with some more challenges of doing field work, not just lugging in a liquid nitrogen tank, but challenges related, to not being able to do field work because of the pandemic and the difficulties in international travel. So I guess, again, just in quick summary, the first collaborative project looks at bats and sequencing their genomes. The second project I'll talk about is taking the perspective of viruses and developing vaccines to stop, the next, outbreak, from becoming another pandemic. And then I'll finish off by talking about some field work in Guyana specifically that has been ongoing over the past decade, but that has obviously been affected by this current pandemic that we're in. Okay, so the first project is on bat genomes and is part of the BAT1K consortium of researchers from around the world. Yeah, so this BAT1K project got started about, I guess, probably at least 2, if not 3 or 4 years ago, and we're still adding on, new researchers. So for those people that are into bats and are into field work, yeah, so the more people we have from different parts of the world contributing to this project, the better, because we found that another one of the challenges is trying to get samples from one country to another country. So a lot of times the country where a lot of the species diversity is, usually that's not where a lot of the high-tech sequencing facilities are. So there's a lot of difficulties with transporting specimens, tissue samples across international boundaries. So we're trying to get more collaborators from different parts of the world helping out with this project. So the objectives of this initiative, the BAT1K, have been separated into 3 phases. So phase 1 is the sequence of species of each of the bat in the 21 families. Phase 2 is sequencing a species from each of the 220 genera of bats. And then phase 3 is the last one. So that's to get the genomes for all 1,400 plus species of bats. So currently we are in phase 1, and the ROM has contributed frozen tissue samples for 1/3 of those families. So I've got pictures of the 7 species or the 7 families of bats that we've contributed to the BAT1K project. So this again involves many universities and museums around the world, but the main DNA sequencing facility collaboration is with the Max Planck Institute in Germany, in Dresden. Okay, so with the outbreak of the pandemic last spring, the Max Planck Institute got funding from the European Union to sequence the genomes of specifically of species in the horseshoe bat family Rhinolophidae, which is, as I mentioned, considered the natural reservoir of SARS-like coronaviruses, but also species in its sister family, the old world leaf-nosed bats, with the scientific family name of Hippocidaridae. So a little bit of background on some of these viruses. So there are 4 types or genera of coronaviruses. So their sort of common or colloquial names are the alpha, beta, gamma, and delta. So the alpha and beta coronaviruses are found in mammals, whereas the gamma and delta coronaviruses are found in birds. So the illustration on the right are of the beta coronaviruses only, so only one of the 4 groups or genera of coronaviruses. So these are the beta coronaviruses. And these are separated into 4 subgenera, including the sarbecovirus, which is in blue at the bottom of this tree diagram. So these viruses occur in horseshoe bats, but also other mammals. So 2 of these sarbecoviruses have caused the SARS outbreak in 2003, and also the current COVID-19 pandemic that we're in right now. The merbicoviruses, so that's in purple at the top of the tree, includes another beta coronavirus that caused the MERS or camel flu outbreak in 2012, which is less contagious disease that was transmitted to humans from camels, with bats as being a suspected reservoir. 1 second here. Sorry. Okay, so as part of this study by Lantine and colleagues in 2020, they also suggested that the horseshoe bat family Rhinolophidae is the ancestral host for all of these beta coronaviruses. Yeah, so you can see some of these other ones are in like the Delta virus, coronavirus is in Pteropodidae, which are the old world fruit-eating bats. But based on this particular study, again, the horseshoe bat family is considered the ancestral host to the beta coronaviruses. So as part of another collaboration with the BAT1K consortium, so we sent in frozen tissue samples for 8 species of horseshoe bats. old-world leaf-nosed bats to the genetic lab in Germany for a specific COVID-19 study. So this will involve sequencing the genomes of these 8 different species, so assembling the 2 billion DNA base pairs, and then annotating all the genes. So this is obviously a very big work, and again, that's another reason why we need the high quality, the high molecular weight, the DNA that you can recover from tissues that are frozen in liquid nitrogen. So from there, we can look at more closely to identify and compare parts of the genome, such as the ACE2 receptor sites, to see how similar or different they are between bats and humans. Also, what genes are involved in the bat immune system response related to tolerance or resistance to pathogens. So these comparisons will give us a better understanding of why bats have a lot of coronaviruses, but typically they are asymptomatic and appear healthy. Yeah, so I think bats will give a good clue on potentially how to get us out of this current pandemic, but also how to prevent any future ones from occurring. Okay, so the second bat project is on virus vaccines and is in collaboration with virologists at Western University. So when the COVID-19 pandemic hit last year, they were just starting clinical trials for a vaccine against the MERS coronavirus. That camel flu related virus that is another beta coronavirus. But then they quickly switched their clinical vaccine trial to the more pressing concerns of SARS-CoV-2, obviously, because there were more deaths happening from SARS than there were from MERS. So their method involved, as you can see in the diagram on the right, involved inserting the coronavirus spike protein gene, so only the spike protein, not the whole virus itself, into a replicating viral vaccine, vector vaccine, as outlined in this diagram. The viral vector develops an RNA that looks like SARS-CoV-2, which initiates an immune reaction from both the spike proteins and the vaccine, but also it enters the cell and replicates more spike proteins that generates an even stronger antibody response. So these clinical trials started last October, and they had favorable results. However, with several large pharmaceutical companies, such as Pfizer and Moderna, et cetera, getting an earlier start on a COVID-19 vaccine, Western has refocused their efforts on using their replicating viral vector system to produce vaccines for potential outbreaks in the future from other possible coronaviruses. That's when they contacted us in November, this past November, to ask about our bat frozen tissue collection from Asia. And this was done through a mutual colleague of ours, Brock Fenton, who gave an earlier CUBES talk, I think maybe a couple of months ago. So the ROM has been doing fieldwork in Southeast Asia since 1993, and more specifically from 2001 to 2007. We've been doing fieldwork in China, where the 2 SARS-like diseases originated. So here's a photo of me in one of the caves we surveyed with mist nets in 2006. I just give an idea of the mist net as opposed to the harp trap I showed you earlier in a picture. So the virologists from Western came to the museum in December to directly subsample from our frozen tissues. So they ended up sampling 211 specimens representing about 56 species of bats from 4 provinces highlighted in the map in the upper right there. Okay, and then in February, a joint media release by Western and the ROM was sent out highlighting the collaboration of our 2 institutions on developing coronavirus vaccines. It got picked up by several news outlets across Canada and also internationally. So far, the tissue samples have been screened for coronaviruses and the RNAs extracted. The spike proteins have been isolated and amplified for sequencing. So the preliminary results are encouraging, and they have come back to the ROM to broaden their sampling geographically to Southeast Asia in general, but also taxonomically to other species of small mammals, such as rodents and shrews, to get a better idea of how widespread and common coronaviruses are in wild populations of mammals. Okay, so now the last part of my talk will focus on the challenges of doing field work during the pandemic. So most of my research is on biodiversity and evolution of bats or mammals in general, small mammals. So I typically take field trips to tropical countries where there's high species diversity. So one ongoing collaborative project is with Operation Wallacea. So they're a British organization that specializes on conservation research by bringing together students to help experts on wildlife projects throughout the world. The annual project in Guyana has been going on since 2011. So basically students learn about field survey methods for not only bats, but also we have experts working on birds, insects, herps. There's a forestry survey. So basically, we get field assistants to help us collect the data. But we teach these students, how to identify bats in my case, but also to set up the bat nets, how to handle bats. So all of the sort of the field survey methodology involved, with doing these types of surveys. So for the bats, we have standardized our sampling grids at 6 sites at Encroachment Forest in central Guyana in South America. So this way comparisons can be made from, among the 6 different sites, but also from year to year. So the objective is to get baseline data on species diversity and relative abundance to enable us to track changes to the ecosystem of Encroachment Forest. So some of the preliminary findings include a surprising amount of variation. So I marked off some of the lows in species diversity, but also the lows in relative abundance. So the red is from 2012. But if you compare that to the highs in 2018, so this powder blue color, you see that there's actually quite a bit of variation. So I was actually even surprised myself. So we had standardized all of our sampling methodology. It's the same 100-meter grid setup we have. We always do it in the summer. Yeah, so I was a little bit surprised that you can get the relative abundance varying by twice as much, and the species diversity can be about 50% between the low and the high. So I was a little bit surprised to see that much amount of variation, even from year to year, because in 2017, so the purple line here, which was, it wasn't a great year, it's similar to the red in 2012, but from 2000, the lows of 2017, the highest numbers occurred in the next year. Yeah, so there is a lot of, I guess, natural resilience in the, the bat fauna that's in Encroachment Forest. So from year to year, things can change quite a bit. So this is one of the reasons why we need to establish a good baseline data set to find out, okay, what is the natural background variation. So we don't want to, make assumptions based on, just a 1 or 2 or 3 year study. Yeah, so the next thing we want to do is we want to start incorporating in other data sets such as weather, but also the forestry surveys that I had mentioned, to see if there's any correlations, between the bat data and these other data sets, and also the data sets from the other organism groups that are being surveyed, such as birds, to see if we come up with explanations on, why there is this type of variation going on. Okay, and then another interesting trend that we found was that there was a 3-year cycle at one of the 6 sites, Turtle Mountain. So the graph on the right, yeah, so these are the 5 most speciose species of bats out of, I think we documented about 55 different species of bats. So these are the 5 most numerous. But you can see with the different arrows here is that that these 3 common species actually disappeared in 2012, 2015, and 2018. So obviously this year, 2021, would have been that 3-year cycle where we wouldn't get these bats, but unfortunately, so this is the 2nd year, so we weren't able to do field work last year, and we're not able to do field work this year. But hopefully we'll be able to get going again in 2022. Yeah, so one of the things that we need to look at here is, there's probably something going on. So these 3 species here in the genus Artibius, they are the dominant food-eating species. So one of the things that we need to look at now is sort of the phenology of plants at that particular study site to ascertain that some fruits are masking, such as figs, which are the main food source for these species in the genus Artibius. So again, we've gotten almost a decade of data. So now we've sort of found out a few trends. There's a lot of background variation, but there's also 3-year cycles going on at least one site. So now we're at the next stage. We want to try to explain, some of the reasons why we see some of these patterns or changes going on. Okay, so I'll just probably end off with conclusions here. So I think I'm getting sort of close to my allotted time, and hopefully there'll leave some time for questions. So some of the take-home messages I want you to get across is first that bats don't transmit SARS-like coronaviruses directly to humans. It goes through an intermediate host that was originally thought to be a pangolin. But even the spike proteins of their viruses are still different enough from the human virus that pangolins probably didn't cause the infection that led to the current pandemic. So we still don't know what the intermediate host is. So still there's a lot of unknowns, in the epidemiology of this particular disease. The second point is that encroachment on wildlife and their natural habitats increases the chance of viruses jumping between different species and mutating before infecting people. So basically animals aren't at fault. In fact, they are beneficial to the ecosystem, as I mentioned, seed dispersers and flower pollinators, such as this picture of this nectar feeding bat feeding on a banana flower. Another point is that bats have a lot of viruses, so that's something we already know, but they are typically asymptomatic and appear healthy. So studying bats and the immune systems will help us in controlling any new diseases that emerge, and also developing we're also working on developing a repository of ready-made vaccines for different types of coronaviruses that will give us a head start on any new outbreaks in the future and preventing them from becoming a new global pandemic. Okay, with that, I'd like to thank you for listening and hope you've got a better understanding of bats and how they are beneficial to the environment and can help in preventing another pandemic. And I'm sure all of us want to get back into the field to be able to continue our research. Okay, thank you.
Thank you very much, Burton. It sounds like you're willing to answer some questions, so I would just open it up to people, and you can either pop questions into the chat or just unmute and ask Burton a question. I'll just open the chat here. Perhaps I could start, Burton. It has always struck me that bats are getting an unfair shake in being accused of being these unprecedented reservoirs of human viruses. And I kind of wonder if the reason that we associate them with it is just because they're so diverse. They're found in so many different habitats that, because Ebola, Marburg virus are also thought to be in fruit bats in Africa, and there are other places. But isn't it the case, for example, that rodents, which are even more speciose than bats, are also important reservoirs of human viruses that cause human diseases? I'm just wondering if they're getting a bad rap, and it's really just by dint of their sheer diversity that we're seeing things like SARS and COVID-19.
Yeah, so I think there are high species diversity, not only in bats but also in rodents. So obviously they are successful from an evolutionary perspective.
Mm-hmm.
Exactly why, as I mentioned, for bats they do have a lot of viruses, diseases, but they appear okay. I think one explanation is also being, I think, proposed for birds, which have other viruses, bird flu, etc. Is that they have high metabolic rates because of flying. So again, that sort of a bird-bat connection is that maybe their immune system is built a little bit tougher, because of all the constraints or difficulties with having to fly from a metabolic perspective. So that might be an explanation. So people have proposed that. I haven't had a good this is the reason why, but people have looked at sort of flight as related to metabolism, which allows them to either resist or tolerate a lot of these pathogens. If anybody else has any other suggestions or ideas, I'd be really happy to hear about them too.
So we have a question from Leslie, and she would like to know how you came to work for the Royal Ontario Museum and what that involves. So you started working as an undergraduate, but you've had a long association with a very good museum. So how did that come to be?
Well, I was doing my undergrad degree at the University of Toronto, and I took the mammalogy course at U of T, and that was taught, the professor was actually the mammal curator at the LOM. So of course, it wasn't on the university campus, it was actually at the museum, which is basically beside the campus. So we were using the collections. So I got to know the curator professor. So I did a couple of projects with him related to a couple of courses, so the mammal course, and also I think it was a systematics course. So his interest, his specialty was bats. So that's how I sort of got into bats. And then when I graduated, I wasn't really sure if I wanted to do graduate work, but he was looking for a research assistant. So I did that for a couple of years and found out I had an interest in that. I liked doing that. Then when the job I have now opened up, I applied for it, got it. But I was also fortunate that I was able to go back for my graduate degrees later on. So I think I sort of did things a little bit differently. So I didn't do the traditional route of, bachelor's directly into a master's and then directly into a PhD. I sort of did it backwards by getting a job after my bachelor's and then going back for my graduate degrees. So sort of a different way to get to the same final point.
Oh, cool. So growing up in Toronto, just an aside, and then we have another question that was just texted to me. Were you fascinated with bats when you were a young person, like a teenager, or is this something that just because of the course in university really struck you?
It was the course. I mean, I loved, actually, I loved Batman, but the campy TV series, Batman. So I'm dating myself now, so I'm actually not a. I like the Batman movies, but not I'm not a huge fan of the Batman movies. So
Well,
some of them are getting a little bit too dark. Yeah, so it's like the total opposite of the sort of the campy Batman TV series. So yeah, so it wasn't until I met up with the curator at the ROM that I started getting into bats myself.
okay. So this is a question from Ying. And so let me just read it here. So we know that the current COVID virus can transmit to many different mammals. Do you think that this could be a concern that the current, so COVID-19 could actually transmit back to bats? In other words, is there a possibility of reverse transmission and actually doing damage to bats in the wild?
So that was a major concern, and I didn't mention it because I didn't do any field work myself, but it came up specifically in the U.S. With the U.S. Fish and Wildlife. So they were trying to keep their bat studies going. So that was one of their concerns, was that what's the possibility of people that are infected with COVID giving it back to bats in North America, where SARS-CoV-2 isn't found. But I think, so I'm not sure exactly what the final decision on that was, but basically the guidelines sort of recommended that, as long as you took the proper precautions with PPE, yeah, so again, wear a face mask, wear gloves, and make sure you wash, all of your equipment after a field trip. To minimize the potential. So again, we don't know if that's a possibility, but really just to minimize, the chances of that happening. So basically, that was the guidelines from the U.S. Fish and Wildlife Service. But I still don't know if there's been any examples. Because I know there's been examples of tigers, like zoo animals. Not a lot, but there are examples of zoo animals getting COVID. So I guess there's always a possibility that the bats might get it, but I haven't heard of any examples of bats in North America testing positive for it.
Okay. This is a question from Oriane. What are the strategies for finding intermediate hosts between bats and humans? So you alluded to the pangolin.
Yeah.
But how is there a systematic search to find perhaps other hosts or intermediate hosts?
Well, I mean, that's one of the things that I'm working on with the virologists at Western. So I think they're still concentrating on bats right now, bat tissue samples. But now I think they're talking about, very soon broadening, the search, for small mammals, again, like rodents or shrews. We have a few other larger species of animals, tissue samples, but a lot of those just come from the zoo. So there are, wider tests that are going on. I'll just give one example from the SARS outbreak from 2003. So they thought like it was a civet. So it's like a mongoose type animal. So basically, they killed all of the civets in the wet markets in China. Literally tens of thousands of them. But then someone actually tested these civets in the wild, from wild populations. I found out that they didn't find any of the SARS virus in the wild populations. So they concluded that the civet cats got infected, but they weren't the intermediate hosts because it seemed like they probably actually ended up dying if they got it. So they were considered the true intermediate host. They did get it, but they didn't find it, any of the wild civets with it. So they didn't assume that it happened naturally. Yeah, so again, from the epidemiologic logical perspective, there's still a lot of unknowns, with COVID.
Yeah. Thank you. Are there any other questions for Dr. Lim? I don't see any hands up or nothing in the chat. All right. Well, thank you very much, Burton, for an illuminating talk. It's a lovely seminar series when you have two bat talks
Yeah.
in the same series in the same year. So really appreciate it. And once again, once the dust is settled on this pandemic, I do invite you to come up and we'll go batting, if that's the term.
Yeah, that'd be great. Thanks for the invitation.
Yeah, thanks so much, Burton.
Okay, thank you.
Bye now.
Bye.